Hi, this is Kumar, Professor of Pharmaceutics, belongs to SNS College of Pharmacy and Health Sciences. Now we are going to discuss about the large-scale sterilization in pharmaceuticals. So we know what is sterilization and what sterilization plays the vital role in pharmaceuticals, everyone knows. So the main purpose of the sterilization is nothing but ensure sterility of the products, equipment, processing and environments. So the indirect role is that it prevents the whole pharmaceuticals industry products as well as the processing. It prevents microbial contamination in the formulation area such as parenterals, ophthalmics, biologicals and active pharmaceutical ingredients. So what are the key requirements needed to perform sterilization in pharmaceuticals? Agent penetration and uniform heat. So there is a need of uniform heat throughout the process. Next, validated sterilization cycles. So each and every cycle, especially sterilization cycle, it has to be properly validated and documented. Next, monitoring with physical, chemical and biological indicators. There is an indicator like physical, it's nothing but a pressure gauge, temperature valve. Chemical is nothing but a purely chemicals like ethylene oxides, formaldehyde gas, and biological indicators nothing but a microbes. So with the help of those indicators, the particular system which is undergoing further sterilization, it has to be evaluated. Next, what are the major equipment types? There are so many sterilizers. So heat-based radiation, gas sterilization system, filtration for thermolabile substances, and specialized industrial sterilizers. Next, first thing we are going to discuss under the sterilizer is nothing but an autoclave. So the principle is nothing but moist heat under the pressure development. The range of the temperature is 121 to 134 degree Celsius. So what are the types of large-scale autoclaves? Horizontal, double door, continuous retort autoclaves and rotatory autoclaves. So the horizontal autoclaves, especially they are meant for bulk loads and large batches. Next, the important thing, continuous retort autoclaves, it is meant for the large volume parenterals. Simply we call LVPs. Next, rotatory autoclaves. It gives an edge when compared to the previous three in the sense of uniform sterilization of sealed containers. So what are the features? We know what is PLC, programmable logical control. Nowadays almost all sterilizers used in the industry, they have the integrated of the PLC and SCADA programming. Next, steam jacketed chambers, vacuum pulsing for air removal for a safety purpose. Next, loading probes and integrated monitoring systems with the high level sensing equipments. So what are the applications? Surgical supplies, glasswares, culture media, water system and large container loads. So the thing what we discussed under the application almost undergoing for the sterilization in autoclaves. Next, hot air oven or hot air sterilizing equipments. So the principle is nothing but high temperature. It delivered the dry, not a moist, the range of 160 degree Celsius to 250 degree Celsius. So what are the equipment features? Like the previous what we discussed, the same SCADA PLC control, integrated sensing control, high temperature resistant construction. Next, rack loading and HEPA filtered air circulation to prevent the passage of the microbes which is so small. Next, uses to sterile glass vials, metal instruments and depyrogenation of heat stable materials. So depyrogenation plays a vital role in the sterilization process. So to remove the pyrogen and it makes the sample pyrogen free. Next, depyrogenation tunnels. Next example, so it's a continuous system. It operates in the range of 250 to 300 degree Celsius attached with the laminar flow. So the application of the depyrogenation tunnels is nothing but high-speed depyrogenation of vials in aseptic production lines. You know vials, especially for the injectables. Next, the very important thing is gas and radiation sterilization equipment. So for gas, the example is ethylene oxide sterilizers. So what are the uses of the ethylene oxide? It's a heat and moisture sensitive materials, especially it applies heat and moisture sensitive materials. The future is the gas mixing and aeration unit, exhaust scrubbers, and it controlled humidity and temperature. It is one of the major advantage, comes under the category of the ethylene oxide sterilizers. It won't build up any humidity, so it makes the sample clear. What are the applications? So catheters, if you want to sterilize catheters, plastics, disposable syringes and filters, which comes under the category of the heat and moisture sensitive products, the preferable method is ethylene oxide sterilizers. Next, gamma radiation and sterilization unit. So gamma radiation comes under the category of the pure radiation. The source of the gamma radiation is nothing but cobalt 60. So what are the equipment? Of course, a radiation chamber. There we kept the products meant for sterilization. Next, source rack, conveyor system, as usual like the all equipments. Next, dosimeter monitoring equipment. How much radiation we are going to give? That is nothing but a dosimeter monitoring equipment. There we arrange, there we make the exact dosage level for the sterilization products. Application. So it applies for the sterilization of medical devices, packing materials and tissue grafts undergoing for the next level of projects. Next, electron beam sterilization, or simply called E-beam sterilization. So what is the speciality of the electron beam? It's a high energy electron accelerators. They have a special equipment and it delivers the electron with the high level accelerations. So through the high energy electron, it gives a rapid sterilization with the high dose. And the one more advantage is, when compared to the gamma radiation, the penetration power is so low. That is the reason they mainly use this electron beam radiation for pharmaceutical packaging, sterilization areas, syringes and medical devices. Next, filtration systems. Of course, we know what is the use of the filtration systems. It's mainly applied in the category of the heat sensitive or thermo sensitive drugs like biologics, example vaccines, enzymes and proteins. They are the highly heat sensitive molecules. As a reason why, to especially for the immunological products like enzymes, vaccines, proteins, whatever may be, it undergoes the sterilization in the filtration category. So here the filtration is nothing but made up of cartridge filter housings and the aseptic membrane filtration with the size 0.22 micrometer. So that is the main property of the filtration systems. So what are the special features? So there is no special features when compared to the other. Here the unique size is filtration only. So under the governing under the stainless steel assemblies, or the entire system is fitted over the stainless steel assemblies, and it has the pressure gauges and integrated testers like a biological indicators, or which indicator is suitable for the evaluation of the filtration system. It has to be attached in the integrated testing unit area. Next, other large scale sterilization equipment. So it may or may not be extensively used in the industry. Based on the industry nature, they will go for the other large scale sterilization equipment. First, hydrogen peroxide vapor, simply called as HPV sterilizers, especially to make the aseptic area of the preparation room. Next, supercritical carbon dioxide sterilization systems is a highly expensive one, especially applied for the thermolabile substances, and previously what we discussed under the filtration unit, biologics. Next, microwave sterilization and continuous steam sterilization.